everybody, welcome to DR.GHOST YouTube channel. Prepping PG entrance one topic at a time. Please like, share and subscribe. Hope you enjoy.
So hello guys, we are now going to talk about an important biochemistry topic which is frequently asked in our exams again. So this topic is about lipoproteins. Okay, what are lipoproteins? Like, since our blood is like watery, it is a fluid and it is polar. So what happens whenever we absorb our fats from our food, it cannot be carried through the polar substance like blood, because fat is non-polar. So we have to find a way to pack the fats in something which is a polar compound, and such polar compounds are called lipoproteins. It means it is made up of lipids and proteins. Okay, so now we'll talk about the structure of the lipoproteins. Okay, you have understood why we need lipoproteins. Now how lipoproteins are polar, and so they mix in the blood and carry the fat. So what happens, its outer core is made up of phospholipids. Okay, the phospholipids are made up of two parts. Okay, the phosphate and the lipids. Okay, the phosphate part is polar, which is outside represented in circles of blue color, and the inner part is made up of lipids, which is represented by blue lines. Okay, so what happens, the outer part becomes polar, which is known as outer hydrophilic core, because the outer hydrophilic core, hydrophilic means water loving, the inner part which contains the lipid part of the phospholipid is known as inner hydrophobic. Hydrophobic means water hating. Okay, hydro means water, phobic means fear, water fearing end. So what happens is, also there will be some proteins embedded in these phospholipids which are called apolipoproteins. Okay, so in the inner side of this structure, lipoprotein will pack non-polar compounds which are hydrophobic compounds like esterified cholesterol and triglyceride. Okay, and in the outer parts, which will pack the polar water-loving parts like unesterified cholesterol. Esterified cholesterol is polar, whereas esterified cholesterol and triglycerides are non-polar. So they will be present in the core. Okay, and in the outer hydrophilic part there will be unesterified cholesterol. This is how we pack the lipids and carry it through our blood. Okay, because fats are non-polar, they cannot be mixed in blood. We need lipoproteins will carry fats through the blood. So now, since we know about what are lipoproteins and why do we need it and what is the structure of lipoproteins, we'll learn about what are the functions of various lipoproteins. So we have different types of lipoproteins in our body, which are namely chylomicrons, VLDL, LDL and HDL. Okay, these are the main types of lipoproteins. We'll study the function of them one by one respectively. Okay, so the chylomicrons. What are chylomicrons? Chylomicrons carry the exogenous triglyceride to peripheral tissues. What happens is, whatever fat we consume in our diet, it's absorbed via intestine and it's packed into chylomicrons. The structure is similar to lipoproteins, and those packed fats or triglycerides are carried to the peripheral tissues via the blood by chylomicrons. That's called exogenous fat because we are taking it from outside world. Okay, in the diet. Now the second type of lipoprotein is VLDL. Okay, very low density lipoprotein we call it. What its function is, it carries endogenous triglyceride to peripheral tissues. Now what is endogenous triglyceride? So I'll explain it to you right now. Okay, see what happens when we take carbohydrate in our diet. 50% of it is converted to energy and the rest 50% is stored. Like 50% is used as energy in our day-to-day life and 50% is stored. Out of that 50%, 10% is stored as glycogen in liver. We all know glycogen breaks down into glucose in fasting state. Okay, and the rest 40% get carbohydrate in diet gets converted to fats. That fat in the liver only by some pathway, and that fat is endogenous fat. So we complain that we don't take fat in our diet, even then we are getting thick day by day. That's because we are taking excess carbohydrates in our diet, which gets converted to endogenous fats, and these endogenous fats from liver are carried by VLDL to peripheral tissues. Okay, so VLDL carries endogenous fat or triglyceride to peripheral tissue. Now the third one lipoprotein is LDL, low density lipoprotein. What it does is, it carries cholesterol formed in the liver to peripheral tissues. Okay, so the cholesterol formed in the liver is carried to the peripheral tissues by LDL, low density lipoprotein. And the last one is high density lipoprotein, HDL, high density lipoprotein. What it does is, it carries cholesterol from peripheral tissues to the liver. So its function is absolutely different from the others. The other three were carrying something to the peripheral tissues. Since HDL does an opposite function from the first three lipoproteins, so the transport by HDL of cholesterol is also known as reverse cholesterol transport, because it transports cholesterol from peripheral tissues to the liver. That's why it is also known as good cholesterol, because it carries the cholesterol which is bad from peripheral tissues to the liver. So now we will talk about various types of lipoproteins and the composition of various lipoproteins. So we have already learned about 4 types of lipoproteins. Now we'll go further into it. So what is the composition of various lipoproteins? we'll study under three headings, which are with the lipoproteins, the lipid present, and the protein or the apoprotein part, which I showed you in the diagram. Okay. So the first lipoprotein is chylomicron. Chylomicron mainly consists of triglyceride, which will be exogenous, which we get from our diet. It also contains negligible amount of cholesterol. So since it is negligible, we do not mention it here. Okay. And the protein part or the apoprotein part it contains is ApoB48. Okay. Now, what does chylomicron do? We have learned that chylomicron delivers the exogenous triglyceride to peripheral tissues. So once it delivers the triglyceride, which is exogenous triglyceride, to the peripheral tissues, a chylomicron remnant is formed. Now the chylomicron remnant, I told you that chylomicron also contains negligible amount of cholesterol in comparison to triglyceride. But since the chylomicron remnant is already unloaded, that is the triglycerides have already been delivered to the peripheral tissues, the cholesterol becomes a significant part. Okay, so the lipid present will be triglyceride plus cholesterol, and the protein or apoprotein present will be ApoB48, which was also present in chylomicron, and also the remnants in the blood uptake ApoE. So in the remnants there will be ApoE. I have highlighted it. ApoE will be present in the remnants. Okay. Now the next lipoprotein is VLDL. VLDL also contains triglyceride, but this triglyceride is endogenous, which was formed from excess carbohydrates we take in the diet. And the protein or the apoprotein part present here is ApoB100. Okay. Now VLDL also delivers the triglyceride to the peripheral tissues. Now when it delivers, it will form VLDL remnant, which contains same like chylomicron remnant. It will contain triglyceride plus cholesterol, and the protein or apoprotein part will be ApoB100 plus ApoE, which it will uptake from the blood. Okay. So in the remnants we can see I have highlighted there will be ApoE protein. Now the next lipoprotein is LDL. What happens when VLDL releases triglyceride? It forms VLDL remnant, also known as IDL, and further it forms LDL. So I have shown an arrow which suggests that VLDL only forms VLDL remnant, which is ideal, and further it forms LDL. So so LDL will contain cholesterol. All the triglycerides are unloaded, and its protein or apoprotein part is ApoB100 plus ApoE. Same thing, because I told you it's formed from unloading of VLDL only. So ApoE will be present in the LDL also. Now the last HDL, which is also known as good cholesterol because it is responsible for reverse transport of cholesterol from peripheral tissues to the liver. It also contains cholesterol, but in the form of cholesterol ester, which was a non-polar one present in the hydrophobic core I showed you in the diagram of the lipoproteins. It is a neutral lipid. Now the apoproteins it contains are ApoA, ApoC, and ApoE. You can remember with the help of mnemonic ACE. Okay. This is all about the composition of various lipoproteins, means the protein and the lipid part each lipoprotein contains. Okay. Now we will talk about the density of various lipoproteins we studied just now. So what happens, the chylomicron has the least density, and as we go on to the SDL, the density increases. Why the density increases? Density depends upon the protein content of the lipoprotein, and more the protein content, that is density is directly proportional to the percentage of protein. I have written here, more the protein content will be there, more will be the density. Okay, and less will be the lipid present in the lipoprotein. Okay. Also, as the density increases, the size will decrease. Means chylomicron will be of larger size and the SDL will be of the smallest size. Okay. So density is inversely proportional to the size. Okay. And density is also inversely proportional to the triglyceride content. I told you, more the density, less will be the fat content. So density is inversely proportional to triglyceride content also. Okay. This is all about this slide. We have to just remember that the density increases as we move towards from chylomicron towards SDL, and the density is directly proportional to the percentage of protein present in the lipoprotein, and hence it will be inversely proportional to the percentage of triglyceride or fat present in the lipoprotein. Okay. And now we should also remember other names of these chylomicrons, or we should also remember the other names of these we should also remember the other names of these lipoproteins. This is based on the electrophoretic mobility of each lipoprotein. So what VLDL is known as pre-beta lipoprotein, and VLDL remnant or ideal, VLDL remnant or ideal is also known as broad beta lipoprotein, and LDL is known as beta lipoprotein. Okay. And HDL is known as alpha lipoprotein or lipoprotein A for alpha and lipoprotein A, which is smallest in size. Okay, just remember these names. These are based on the electrophoretic study. LDL is the beta lipoprotein, HDL is the alpha lipoprotein or lipoprotein A. Now VLDL will be pre-beta lipoprotein, and the remnants will be, or ideal will be, broad beta lipoprotein. This will be also helpful in
hyperlipoproteinemias, which is a disorder. Okay. Now since we have learned in detail about the anatomy and what physiology of lipoproteins, how they work, what are they made of, and now we'll learn about an important disease based on these lipoproteins. Okay, that is Tangiers disease. What is Tangiers disease? Tangiers disease is also known as familial alpha lipoprotein deficiency or hypoalpha lipoproteinemia. Okay, what it means that there will be alpha lipoprotein deficiency. I told you to remember the other names of HDL and other lipoproteins. What was HDL called? HDL was known as alpha lipoprotein or lipoprotein A. So there will be deficiency of lipoprotein alpha or lipoprotein A in the Tangiers disease. Now we know what was the function of HDL. It was doing reverse cholesterol transport from peripheral tissues to the liver. Now since the HDL will be low, which is due to a mutation in a gene called ABCA1 gene mutation. You have to mug up this. there will be decreased HDL. Now, since there will be decreased HDL, the cholesterol ester which will be accumulated in various peripheral body tissues. Like one of the common organs is tonsils. So cholesterol will be deposited, and the tonsil will give orange to yellowish color due to cholesterol deposition, since HDL cannot carry the cholesterol from the tonsils, that is peripheral tissues, to the liver because of low quantity of HDL. Okay. Also, this cholesterol will be accumulated in liver, spleen, causing hepatosplenomegaly. In lymph nodes, there will be enlarged lymph nodes, and also it will be accumulated in Schwann cells. Schwann cells, this will lead to peripheral neuropathy. This is the reason of peripheral neuropathy in Tangiers disease. So, whenever you find a question talking about tonsils, large yellow tonsils with peripheral neuropathy, hepatosplenomegaly, your diagnosis should go towards Tangiers disease, which is due to alpha lipoprotein or HDL deficiency or decreased amount. Okay.